We're really pleased to welcome Dr. Rogan Tokach, who's a new assistant research professor working on applied honeybee research at North Dakota State University. Rogan graduated with his PhD in June of 2025 from Auburn University, where he studied under Dr. Jeff Williams. His research at Auburn focused on developing sustainable parasitic mite management strategies for beekeepers. His work involved finding alternative treatment options for Amitraz-resistant varroa mites. He's also had the opportunity to travel to Thailand to conduct research dedicated on filling knowledge gaps concerning Tropilaelaps mite monitoring, detection, dispersal, and management. Rogan completed his MS degree at University of Nebraska-Lincoln under Dr. Autumn Smart and Dr. Judy Wu Smart in 2022, where he studied the impacts of pesticide contaminated environment had on honeybee colony functions. Rogan has 15 years of beekeeping experience as a hobbyist, research, and commercial levels. Thank you very much, Rogan, for joining us. I appreciate you with the time difference joining us. Welcome. I'm going to turn it over to you, and you can start sharing your screen and begin whenever you'd like.
Yeah, well, first of all, thank you so much for having me. And let me get all my things in order on Zoom. So I'm really excited that you guys have allowed me to speak, and I'll be speaking on the infamous Tropilaelaps mite. And as you can see on the right, I have a couple different logos. Of course, now I'm at North Dakota State University, but a lot of this research was conducted at Auburn. And if you couldn't see, I'm going to start this video over again, and hopefully you see a small little mite running around on that frame in that screen. And that is that mite that we're going to be talking about today, the Tropilaelaps mite. So one of the things that I always like to start out with when I talk about Tropilaelaps mite is I like to start out with a little bit of background on varroa mites. And so I'm sure it's not going to be a shock to you that varroa mites are a consistent leader of colony losses at the backyard, sideliner, and commercial beekeeping level. Doesn't really matter what level of beekeeper you are, you're having problems with varroa mites. And a lot of beekeepers do have problems with varroa mites and have that background of varroa mite knowledge. However, I want to take you back little over 2 years ago to January 2023. This is when I started my PhD. And there was a new mite kind of bursting onto the scene, and beekeepers kind of got a little bit of a wandering eye to this mite. Now, I want to point out my advisor at Auburn didn't really like this slide, but he's no longer my advisor. I've since graduated, so I can use whatever slides that I want. But as you can see, Tropilaelaps really kind of took the beekeeping industry by storm. There was a big push for a lot of Tropilaelaps research. My advisor, having previous work done with Tropilaelaps, having contacts in these different areas around the world, gave me the opportunity to be able to turn some of my PhD work, which was going to be solely focused on Varroa, to looking at a new parasitic mite with Tropilaelaps. So as I kind of mentioned, I like to talk about the similarities and differences between Varroa and Tropilaelaps because they do have some of these similarities, and beekeepers have a really good background usually with Varroa mites. So to start out with some of those similarities, they're both parasitic mites on Apis mellifera, which is that western honeybee that we all manage. They both reproduce within the brood, and they're both capable of vectoring viruses, or serving as a vector and transmitting these viruses. Now, I want to point out we know a lot more about the viruses that Varroa can transmit compared to Tropilaelaps, but we're working on that research for Tropilaelaps. Now, after that, we start to see some really distinct differences. Varroa mites can feed on larvae, pupae, and adult honeybees. Tropilaelaps can only feed on larvae and pupae. They cannot feed on adult bees, and that's a really important thing to know. Another interesting thing is that varroa mites require a short adult bee feeding phase post-mating before they're capable of laying eggs. Now, what that means is that after a varroa mite, a foundress mite, enters the cell, lays her daughter mites, those daughter mites do sibling mating. Those daughter mites will then emerge, and they typically have to feed on adult honeybees for 5 to 7 days before they go back in and begin reproducing themselves. Now, Tropilaelaps, since they don't feed on adults, don't have the same post-mating feeding phase requirement, so they're capable of exiting the cell as a daughter mite and going in usually within 48 hours and reinfesting a new cell. And this allows Tropilaelaps to reproduce quite a bit faster than varroa mites. Now, varroa mites, another thing that harms they're able to reproduce as fast as Tropilaelaps is varroa mites have a 30-hour egg laying interval compared to Tropilaelaps, which is just 24. And interestingly enough, when found to be co-infesting the same colonies, it was shown that varroa mites had 50% non-reproductive females compared to 30% for Tropilaelaps. So what this really means is that in similarly infested colonies, Tropilaelaps was outcompeting varroa mites. So here are these two mites side by side. You can see that Tropilaelaps is about the same length, but it's 1/3 of the width. You can also see that it has much longer legs, and you'll see in some of these videos that helps the mite move around much quicker on the frame. Whereas varroa mites generally rely on the bees to move them around in the colony, Tropilaelaps is very capable of moving around on its own. So I want to point out that Tropilaelaps, Apis dorsata and Apis laboriosa, which are giant honeybee species, are believed to be the original hosts of Tropilaelaps. And so what that means is that the original host range of Tropilaelaps is generally where these giant honeybee species were. So this is kind of what the original host range we think that Tropilaelaps looked like. I also want to point out overall, there's 4 species of Tropilaelaps, but really we're only concerned about one species, with that being Tropilaelaps mercedese. Out of the 4 species, only 2 have been found to parasitize Apis mellifera, which is that western honeybee. And of those 2, only Tropilaelaps mercedese has been found outside that host range. So as you look on this slide, it's really important to note that only the Tropilaelaps mercedese species is the one being found outside that range. So when I flash, this is the kind of native range to that giant honeybee species that we consider. As you can see, that Tropilaelaps mercedese species, it's being found outside. I also want to point out my source for this. That's 10 years old. And so that's not exactly up to date as to where Tropilaelaps mercedese is currently being found. So when talking about Tropilaelaps, it has a very similar development stage to Varroa, where it goes from an egg to a 6-legged larvae to a protonymph, a deutonymph, and then it has your adult female and your adult male. One of the interesting things, though, is that the adult females and the adult males look almost identical in the colony, and the adult male, unlike that of Varroa, can survive outside the cell. Typically, the female lives for about 50 days, whereas the male lives for about 5 days. But you really need to have a microscope to be able to differentiate the females from the males. It's also important to note that Tropilaelaps reproduce similarly to Varroa, but they reproduce a little bit faster. If you remember, I said that Varroa mites typically will feed on adult bees for 5 to 7 days before they'll begin reproducing themselves. Tropilaelaps, of course, doesn't have that same requirement, so they're going in generally in less than 48 hours and reinfesting new cells, allowing them to reproduce quicker than Varroa mites. It's also interesting to note that Tropilaelaps feeds on multiple sites on the developing bee, whereas Varroa mites will feed on one wound and just keep opening it over and over and over again. With this picture on the right of that screen, you can see that all these different little dots on that picture, those all represent different feeding sites for those Tropilaelaps mites. And so what this can cause is this can cause different deformities depending on where those mites are feeding while that honeybee pupates. It can cause deformities of maybe legs or antennae that Varroa mites typically don't cause unless they're vectoring something like a deformed wing virus. So one of the best papers that actually shows how Tropilaelaps impacts honeybees is coming from this paper out of Thailand that actually my advisor was on. And it shows that Tropilaelaps feeding reduces some honeybee physical forms. So what they did is they had 3 different treatment groups, and I'll take you through each different treatment group. They had their CC treatment group, which was essentially just a brood cell that they didn't do anything to. It's just a capped brood cell. They had their IC treatment group, where they made a small incision on one of those brood cells, but then they didn't do anything else. They allowed the bees to seal it back up, and then that was just another one. Then they had an IT. They made a small incision on that brood cell after it was just capped, allowing the bee to pupate, and they inserted a Tropilaelaps mite in there. Now you can see that with the inserted Tropilaelaps mite, the weight of the newly emerged bees was significantly lower when that Tropilaelaps mite was in that cell. That's not really shocking. We typically expect if that bee is being fed on during pupation, naturally would have a lower weight at emergence. Another thing that they proved was that the proportion of deformed bees was higher when they had a mite. Again, something that we would typically expect if you have a Tropilaelaps mite in that cell while that bee is pupating, feeding on that bee, it's causing increased deformities. And then lastly, and most importantly, feeding reduced the honeybee longevity. So that one, those cells with those bees that emerged that had the Tropilaelaps mites in those cells, they had a lower overall survival rate compared to the ones that were in the control cells. So that just kind of gives you a little bit of a background on how Tropilaelaps mites are damaging bees in these colonies. Now, you may be wondering, where is this mite currently? Well, one of the chapters for my dissertation was really focused on finding out where this mite was. So what I did was I combed all the peer-reviewed literature, I went through everything and found, okay, where has this mite been either morphologically confirmed by keying the mite out, or where has it been confirmed through DNA evidence? And what I found was a map that kind of looked like this. The other thing that I want to point out on the map is that we have this key for associated species. I was really interested to see, was this mite found in association with Apis mellifera, that western honeybee, Apis dorsata, the giant honeybee, both, or if they didn't specify at all. And so it's not really shocking to see that a lot of the ones that we see are, yeah, it's been confirmed in association with both species where the Apis dorsata is native. But as you can see right here. But you can also see, as you zoom out, look at that region off there into Eastern Europe and Western Asia. These are new conformations that just happened in 2024. So it's important to note that Tropilaelaps mercedese is really expanding its overall geographical range. And I've heard that it's been expanded even further than this, although those are unconfirmed reports. Another really frustrating thing is that it was found in Russia in 2022, but we didn't get any papers or any confirmation of the mite until 2024. So that's allowing these mites to potentially go even further until we know that some of these regions have Tropilaelaps. So what we're trying to do as a research community is come up with better overall monitoring and detection techniques and then have more rapid detection to be able to confirm Tropilaelaps in new areas before it becomes a problem and has the potential to spread into other areas. But of course, when you're talking about spread, you may be wondering, how is the mite really spreading? Well, when I started my PhD, we really didn't have a lot of knowledge as to how Tropilaelaps is spreading. We thought that it was spreading similar to varroa mites, where it was either beekeeper mediated dispersal, where beekeepers are moving infested colonies around different countries, and then they're moving frames between infested colonies and non-infested colonies. They have the mites found within the brood, and that's how Tropilaelaps is moving around. Or it could be something through potentially drift or robbing behaviors, where a really high infestation colony might be getting robbed out or have bees that are leaving and exiting the colony with Tropilaelaps mites on them, and then they go into a new colony, or that weak colony gets robbed out by a stronger colony and the mites hitchhike back. Now, the issue is that no one had ever confirmed Tropilaelaps mites on honeybees exiting the colony. And so that's what I wanted to do with some of my research. So here I am. I like to say a young, naive researcher, although this was like a little over 2 years ago, so I don't know how much I've matured. But this is me fresh off the plane, looking to solve all of the Tropilaelaps problems in one summer. And here's where I was doing my research up in northern Thailand in Chiang Mai. And I'm very happy because I just got all my supplies through customs, which I was extremely worried about. So one of my projects was I wanted to establish funnel traps to study Tropilaelaps dispersal. And so my goal is I wanted to see if I could capture adult honeybees exiting the colony and then alcohol wash them and see if I could find just a single mite. Because if I could find just one mite, then I could say it's plausible that as adult bees exit the colony, they have a mite. That bee could then drift to a new colony, and all of a sudden, wow, that's one way that these mites could disperse. So this is the paper I always like to show. These are what the funnel traps are supposed to look like. Unfortunately, I was dealing with what I could get through customs, so we had kind of the Walmart Great Value version of these funnel traps. But I will say that they did prove to be effective. So here I am installing all of these traps on 6 colonies, and then this is just typically what the trap would look like. I put a cup in, I just allowed bees to try to exit the colony. I took them, collected them, alcohol washed them, and like I said, I was looking for one mite. So I did this in July and August of 2023. I washed 7,250 bees and I found 0 mites. So a little discouraging, but what I really want to draw your attention to is look at that average Tropilaelaps cell infestation number. We were dealing with 0.75%. That means that less than 1 in 100 cells on average in these colonies had a Tropilaelaps mite in there. That meant that we had really low infestation colonies. And so the thought would be these mites aren't feeding on adult bees, so why would they need to be on the adult bees when they're exiting the colony? This is a risky behavior for a parasite. It's much less risky to try to go and feed on developing brood where you know that you have a food source. So I went back in January of 2024 and I thought, okay, I'm going to find the highest infestation colonies that I can. I'm going to throw some traps on them and we're going to see what can we find. And lo and behold, we found mites exiting on adult bees. So when I show you this table, I washed about a third of the number of total bees, but you can also see I had really, really high average Tropilaelaps cell infestation numbers at 25.5%. This means that where I was less than 1 mite per 100 cells, now about 1 in every 4 cells that I open had a Tropilaelaps mite in these colonies on average. So that's really high infestation. So I proved that, yes, we don't see this behavior at low infestations, we see it at high infestations. Is there an equilibrium where you start to see this behavior occurring? Well, my colleague, Dr. Dan Arel, who's a postdoc at Auburn University, he went over to Korea to look at some of this research as well, because we were trying to do some research in Thailand and Korea, 2 different climates. And here's kind of what we expected. We expected to see that, yes, as you increase the number of infested cells, then you start to see that you will see Tropilaelaps on exiting on adult bees. But we expected to see that behavior much lower on Varroa at lower overall brood cell infestations because we know that Varroa mites are being found on adult bees because they're constantly feeding on adult bees and we can use the alcohol wash as a method to detect them. What we found was much more interesting that there wasn't really a huge difference between when we started to see this behavior. What Dan found when he put these traps on is that as low as 5%, he found Tropilaelaps on exiting on adult bees. And so what that means is that at just 5% cell infestation, you can see this behavior where Tropilaelaps can be found potentially dispersing using adult bees, which was really interesting to see and not really what we thought. And I want you to do is keep that 5% in mind because it's going to come up a little bit later in the talk. Now, as we transition a little bit, I'd like to talk about Tropilaelaps monitoring. Tropilaelaps monitoring is near and dear to my heart. It's actually my favorite overall thing to talk about when considering Tropilaelaps. And what I like to always start out with in Tropilaelaps monitoring is a little test for you of can you spot the Tropilaelaps mite that's on the sticky board? I also want to point out this is a two 1 inch by 1 inch squares, so you're dealing with 2 square inches of sticky board. I'll give you about 5 or 6 more seconds. And it's right there. That's what you're looking for when you're looking for that Tropilaelaps mite. So now I'll give you guys another chance. You know what you're looking for. How about this one? Completely new photo. And if you saw multiple, it's right. It's a trick one. I was a little bit mean to you. There's actually 6 total mites on this sticky board. You can see them all here. And then that bottom, that farthest one down at the bottom, that has 2 mites in that circle. And just for comparison, that's what the Varroa mites look like. So I always like to show this just to show how difficult these mites can be to spot. And the other thing I like to point out is that these mites are commonly confused. A lot of times when I give this talk at different conferences, I'll have a beekeeper come up and say, I found this on my sticky board. I think it might be a Tropilaelaps mite. Fortunately, it's never been a Tropilaelaps mite, but we have had some interesting ones that people have shown me. A lot of times what people show me is what's pictured on the right, which is a male varroa mite. People can sometimes confuse that with what a Tropilaelaps mite looks like. On this picture on the left, you can see a varroa mite. This is a bee louse or a brawlifly also pictured, which has those longer legs. On the far left, that rounder mite, that's a pollen mite. And then down on the bottom, that's the Tropilaelaps mite in that photo. The other thing that I like to say is who knows what you might find in your colonies. While I was looking at some sticky boards over in Thailand, I found this. Now I knew it wasn't a Tropilaelaps mite, but I was like, what the heck is this thing? Apparently, it's an Afrocypholaelaps mite, which I don't even know exactly what that is, but I think it's some form of pollen mite. And the other thing, here's another one that I found, someone found for me. So this was presented to the New Hampshire apiary inspector, and so you can see on this video, they are moving around a lot like what Tropilaelaps mites might look like. This is probably the closest someone's ever been when showing me. And I'll try to play that video one more time so you can see those mites running around. But I always like to say this is what I really enjoy about beekeepers is that they're being very cognizant, and this is what you want to do. If you see something very interesting, call your local apiary inspector, take a really nice video, try to get it as clear as possible because it is really difficult to see with graininess, and send it off. And who knows, maybe you'll have that come sample because we really want to try to have early detection. And so I love when beekeepers talk to me about these because it shows that they're really interested in what's going on in their colony and they're being extremely observant. So now as we transition a little bit to a different form of monitoring, these are the kind of 5 most common Tropilaelaps monitoring methods. You have the alcohol wash and the powdered sugar shake, which are of course adapted from varroa mites. The bump test, which I can talk about a little bit. The bump test is a test where you take a frame that has open larvae on it, preferably more 4th or 5th instar larvae, and you take it, you put it over a metal pan, and you wrap it 2 times really hard on each corner. So a total of 8 times. You then take all that contents, you wash it down with water, you strain it through something, a really fine sieve like cheesecloth, and then you take that cheesecloth and you inspect it for Tropilaelaps mites. The other ones that you can do are brood uncapping. So of course, uncapping individual cells of brood, and then mite drop or using sticky boards. So the best overall paper looking at some of these comparisons was done by Dr. Jeff Pettis in 2013, and he was looking to compare some of these methods and see which one was the most efficient, but also which one had the best detection rate. So this is a table that has been modified from his results, but you can see the sticky board had the highest infestation verification at 81.3%, and worker brood was about even at 75.7%. Now the bump test was pretty okay at 50%, and the alcohol wash was doing very poor under 10%. Now what he decided was that the bump test should be the recommended method. Why is that? Well, it really comes down to the time per colony to do it. Uncapping 100 cells is a lot of work and it takes a lot of time. And sticky boards take multiple times and travel to the apiary to be able to put that sticky board in, take it out, and then go through the entire sticky board. So what he found was kind of the bump test is giving you the best bang for your buck, and that's the one that's currently being used by most apiary inspectors. So when we were over in Thailand, Dr. Dan Arell and I, we wanted to say, okay, let's see, is Dr. Pettis' test correct? So this is all our raw data from our Thailand monitoring results. This is the Tropilaelaps cell infestation, and we had up to 27, all the way from 0 to 27, our sticky board numbers. You can see at the bottom, this is our percentage detection. So 92% for Tropilaelaps cell, which is brood uncapping. We had 100% detection for sticky board, 73 for bump, so pretty good. And then only 38 and 35, that would be for the alcohol wash and the sugar shake. And what's also important to note is that we didn't start seeing consistent yeses until about that 5% brood cell infestation mark. And so what Dan did was really cool is he wanted to see what was the DT50 or the detection threshold where we see 50% detection where on the Tropilaelaps cell infestation. So what does the brood infestation have to be to detect at a 50% confidence rate? And this is kind of thinking along the lines of something like an LD50 or an LC50 when you're thinking about, okay, at what level or dosage does 50% of the population die? We wanted to see at what brood infestation do we have a detection threshold at 50%? Are we 50% confident that we can find it using the given test? Well, what we found was that at that detection probability of 50%, it was right a little bit above 1% cell infestation or brood cell infestation of Tropilaelaps that the bump test gave us that DT50. It wasn't until over 5% for the powdered sugar shake and alcohol wash. Now, if we think back to that 5%, we started seeing mites exiting colony at around 5% cell infestation. And so that's important to note because if we rely solely on powdered sugar shakes and alcohol washes to do detection for Tropilaelaps, we might see that these mites are already capable of dispersing into new areas by the time we might even detect them. The other problem, though, with this is that there's really no perfect monitoring method. All these are really relying on physical monitoring and physically seeing that mite, which, as you can kind of see, is really difficult to do. So that's brought up our question, what about using eDNA? So one of the projects that Dan and I have been working on is trying to find what sample would be the best to collect eDNA for Tropilaelaps so that beekeepers don't have to rely on physically seeing that mite. If we have something pops hot for Tropilaelaps, we can go do a whole bunch of sample collections and send them all in for eDNA sampling and be reasonably confident that we found that mite. So that's something that we're really working on, figuring out what samples work the best and at what detection threshold, what cell infestation can we actually find it to work. Some of the other things that we're doing is we're really working with some of these different groups for some preemptive action occurring in the US. So we're trying to train apiary inspectors. We took apiary inspectors over to Thailand to work with Tropilaelaps themselves. We're working with extension specialists, and then we've been trying to work with other beekeeping leaders, such as organizations like American Bee Federation, American Honey Producers Association, and I'll actually be going to California State Beekeepers Association, and we'll have a booth. Auburn will have a booth there where we'll be showing mites and allowing beekeepers that attend that conference to see what it looks like in person to try to find mites on these sticky boards and kind of share what some of our resources and results have been. So now I'd like to kind of transition to the last main portion of my talk, which really focuses on Tropilaelaps management. We've talked, okay, maybe we try to detect it. What actually kills this mite? And that's some of the research that I've been trying to do. And so what I like to point out first is that Thai beekeepers have been dealing with Tropilaelaps since Apis mellifera was introduced. Apis mellifera was introduced into the region in the 1960s, and they've been having to deal with Tropilaelaps ever since. So Thai beekeepers really use a mix of cultural and chemical controls to help manage Tropilaelaps. One of the most interesting things that Thai beekeepers use is since we don't have a great detection threshold or way to detect Tropilaelaps, they use bald brood as their way of seeing if they have high levels of Tropilaelaps. So whereas for us, we go out to see if we have high varroa infestations and do things like alcohol washes, one of the things that they try to use to detect is looking at those bald broods, so those uncapped cells with those purple-eyed pupae. And I talked to one beekeeper that said if he has over 30 cells of bald brood on a frame, he knows that he has a high Tropilaelaps cell infestation and he needs to do some form of treatment. When you actually look at some of the treatments they do, one of the important things to note with Thai beekeepers is that they keep really small colonies. I never went into a colony in Thailand that was over 8 or 9 frames. They were keeping them really small. And one of the thoughts on that is because if you don't have large brood areas, that really limits the Tropilaelaps mite ability to rapidly reproduce and build up in high numbers. The other thing is that they use this And shoot, I had somebody tell me what it was, and I'm blanking on the name of it, but this is what they have when you go to a flower arrangement. It's the foam that they stick in. That is what they're dipping. They're dipping that in formic acid, and that's one of the ways that they're using to treat Tropilaelaps is using formic acid. They're also using synthetic chemicals. I believe the top one is flumethrin. The bottom is a fluvalinate strip, so something that has been used against Varroa in the past. These are strips very similar to things like Apivar, where they're just dropped into the colony and allowed to kill the mites. So when we're talking about what has been tested for Tropilaelaps to use, most of what's been tested is some kind of modified Varroa mite treatment. So they're really looking at seeing, okay, well, if it works on Varroa, does it also work on Tropilaelaps? So you have things like organics, like formic acid and hops beta acids that have been tested, which is HopGuard, of course, and Formic Pro. Cultural with its hygienic behavior and a brood break. And then synthetic, which is amitraz or apivar. Fluvalinate, which you might be more familiar with, is apistan, flumethrin, and then Coumaphos, which is more like Checkmate Plus. So this is the best overall publication that really talks about some of these different things that have been tested. This is a 2017 paper by Dr. Jeff Pettis. And what he did was he tested a few of these different treatments against Tropilaelaps. And so on the y-axis, that's the number of infested brood cells. So that's just the brood cell infestation out of 100. And on the x-axis is the number of days. And so he did this in both the rainy season and the dry season. And so that first one on the left, what you can see is that formic acid and sulfur performed really well. They were lower, one mite or lower, and that amitraz was actually the worst one. So one of the most effective things against varroa mites was not effective against Tropilaelaps mites at all. And one of the hypotheses that we have with that is that Tropilaelaps mites, since they're not out on the adult bees very long, they're not outside the cell, you really need something that's going to be able to penetrate those cell cappings and kill the mites within the brood, or rapid acting treatments. That's why we think formic acid is such a good treatment, is because it's capable of penetrating those cell cappings and killing those mites in the brood. It doesn't rely on contact like something like amitraz does. Now, Dr. Pettis did it again in a different season and found yet again that formic acid performed the best, but not quite as well in the original study. The other important thing to note is that sulfur performed okay in the first study, but sulfur had a very negative effect on colony strength, and those colonies were really succumbing to having sulfur just dumped into their colonies. The other thing that is really important to show is that brood breaks have shown a very strong and viable treatment method against Tropilaelaps. Now, that shouldn't come as a shock to you. We know that Tropilaelaps mites are incapable of feeding on adult bees. The other interesting thing is that Tropilaelaps mites do not feed on honeybee larvae until that larvae is roughly 3.5 days old. So if we think about general honeybee biology, if you remove all of the brood, all of the larvae from the colony, and then allow the queen to start laying, that queen will lay. It takes 3 days for that egg to hatch, and it takes another 3.5 days for that Tropilaelaps mite to be able to feed on the larvae. The longest we've ever found a Tropilaelaps mite in some kind of lab study to survive without having access to larvae is somewhere between 5 and 6 days. So in theory, all of the mites should be dying. Now, we found it to be an extremely viable treatment, but we've never seen 100% efficacy for the brood break. But we have seen efficacies of over 95 to 98%. So this is proving to be an extremely effective treatment against Tropilaelaps because these Tropilaelaps mites are starving without having access to larvae or pupae within the colony. So here's just kind of a breakdown of all the literature that's showing some of the current tested treatments, and they've really had mixed effects or mixed success against Tropilaelaps. Things that have been effective, again, is that formic acid capable of penetrating those wax cell cappings and killing the mites in the brood, and of course a brood break. Some things that have been moderately effective are more of the synthetic treatments like flumethrin, fluvalinate, and Coumaphos, which are being put into strips. And some things that are not effective are amitraz and those hops beta acids like HopGuard. Those have proven to be not effective against Tropilaelaps, relying on contact and not being fast acting enough to kill the mites. So finally, I'd like to really cover some of my research experiences in Thailand and doing some of the cover some of the projects that I've been working on, and one project in particular. So I was being funded by APHIS, which is the Animal Plant Health Inspection Service, which is really interested in eradication of Tropilaelaps. Because what we're trying to avoid is we're trying to avoid history repeating itself. This is a figure showing the spread of varroa mites. What's important to think about varroa mites is within the first detection of varroa mites to 10 years, it was found within a decade, it was all over the US. Varroa mites spread extremely quickly due to that migratory nature of the commercial beekeeping industry. So it didn't take long for varroa mites to be found once, they were found all over the US. And so what we're trying to see is can we avoid that with Tropilaelaps? Because there's a huge worry about Tropilaelaps introduction because we don't have a current eradication strategy. As I've been told by APHIS, they'll treat this on a case-by-case basis. So what I'd like to kind of propose to you is a worst case scenario. So not meaning to pick on you in California, but just thinking what happens if that mite comes in during the almond pollination in California when there's millions of colonies in that area. Well, all of a sudden, those colonies will disperse, and you'll have the mite up in Washington. You'll have it down in Florida. You'll have it, I'll be dealing with it up in North Dakota. It'll be on the East Coast, and pretty soon that mite is everywhere. And at that point, when that mite has reached everywhere, you are no longer looking at just eradication. You are now looking at management because that mite is everywhere. You're not going to be able to eradicate it anymore. But APHIS is really interested in seeing, well, what happens if we get an early detection, we eliminate that first incursion of the mite? Do we have a treatment or management plan ready so that we can treat all these colonies potentially in a quarantine zone and be reasonably confident that we killed all the mites in that area and then we'd hopefully be able to eradicate that mite. So that's what some of the research that I came over in Thailand to do. And what I really wanted to do was to build on the basis of a brood break. We know that brood breaks are extremely effective for Tropilaelaps, but all the literature shows that they're not 100% effective. And remember, if we're wanting to do eradication, we need every single mite to die. So what I wanted to see is can we take that backbone treatment plan of a brood break and add on some form of chemical treatment to try to get that 100% efficacy, eliminate all the mites within the colony. So to do that, I set up 4 treatment groups. I had a brood break only treatment group, a brood break plus formic acid where I used Formic Pro, a brood break plus oxalic acid dribble, and then an untreated control. So I bought colonies and initially assessed them. I bought colonies from a local beekeeper and assessed them for adult population, capped brood area, and Tropilaelaps infestation. I assessed them for Tropilaelaps infestation through a couple ways. One of them was uncapping 200 cells per colony, and the other one was by using sticky boards periodically. I then, on day 0, caged all of the queens in my brood break colonies, and I released them 24 days later. Why 24 days? Well, if we think again on honeybee biology, it takes 24 days for a drone to develop. So my thought process was if a queen had just laid a drone egg, I waited 24 days to release her, well, then that drone should have emerged and there would be no brood left in the colony. I then also on day 24 put my treatments on. So you can see this is the Formic Pro pad. I only did one half dosage of Formic Pro, which is 1 strip for 10 days. And then I did my oxalic acid dribble. After 60 days, I again assessed colonies for colony strength and looked at Tropilaelaps cell infestation on day 60. The other thing that I want to point out in this picture is that no matter where you are in the world, the undergrads have to help you out with all of your grunt work in the field. So very thankful for all the assistance that I got while I was over there. So I'm not going to kind of bore you with some of the colony strength metrics because we didn't see huge differences. A slight dip in the brood break plus formic acid due to formic acid being used at high temperatures. But what we found was really interesting results for Tropilaelaps infestation. So on the y-axis, that's showing the percentage of infested worker brood cells, and on the x-axis, you can see day. And what I was really concerned about is that we had extremely low beginning cell infestations, less than 1% across the board in all of our treatment groups. But by day 60, we shot up in the control colonies with no treatment. They were showing 15% cell infestation. The other important thing that I wanted to note to you is that on day 60, you see we don't have any orange circle or orange bar. That's because we didn't find a single mite in any of our brood break plus formic acid colonies when we uncapped the brood. That's what we wanted. We didn't want to see any mites. We wanted to see if we could try to eradicate these mites in these colonies. So then, as you move on, this is looking at the mite fall on sticky boards. That's what's being shown on the y-axis in 24 hours. And then on the x-axis, again, day. You see a similar trend where you see some mite fall picking up early on as those mites are starving in the brood break colonies on day 12. Day 25, you're picking up some stragglers. On day 50, you see that spike in those control colonies that don't receive any treatment. Again, 0 mites being found in the brood break plus formic acid. Very encouraging. However, on day 60, we did find a few mites in that brood break plus formic acid colony treatment group on some of those sticky boards. And so what that meant is that none of our treatments were 100% effective, which is what we wanted. So what we proved is really that brood breaks alone remain an extremely effective method for treating for Tropilaelaps mites. All you really need is a brood break, and you're going to wipe out that Tropilaelaps mite population and get over efficacies of over 95. We had efficacies of 97 to 98%, even in the brood break alone. And we found that that additional chemical treatment, while maybe slightly increasing our overall efficacy in killing more mites, it didn't eliminate every single mite from these colonies, which was a little disappointing to see. But what's important to say is that mite drift could have impacted these results. As I've shown to you earlier in this presentation, mites can drift in between colonies. We housed all these colonies in one location. It was plausible that some of our control colonies were reaching really high infestations, coming down and crashing, and that colonies were being reinfested in the area, even though the treatments had been effective. I do want to point out that we had these control colonies that it only took 60 days, and we had some of them crashing and dying because of these really high Tropilaelaps cell infestations. So I went back last year to Thailand, and I wanted to see, okay, well, what happens if I treat the entire yard? If I do all the colonies with the brood break plus formic acid, does that give me 100% efficacy? And I'm still working through that data right now and hoping to publish that in the future. Lastly, I kind of want to leave you with one of the most interesting graphs that Dr. Rel put together. So this is Tropilaelaps brood infestation over time. He took the reproduction rate for Tropilaelaps from my research in Thailand, which is the dotted bar, and from his research in Korea, which had a little bit slower of a reproductive rate. And he also looked at the Varroa reproductive rate. What's interesting to note is that the brood infestation, it really takes 4 months in Thailand and 5 months to start really spiking in Korea. So you can really see the differences between the reproduction rate of Tropilaelaps compared to Varroa. Tropilaelaps is capable of reproducing at a much faster overall rate than that of Varroa, and so they can overtake colonies in a shorter period of time. The other important thing that I want to point out, though, is that in Thailand, Tropilaelaps is the predominant mite species. Thai beekeepers have to deal with Tropilaelaps more than they have to deal with Varroa. Tropilaelaps has outcompeted Varroa because there is brood all year round. In Korea, there is less brood year round. It has more temperate climates. Because of these temperate climates, they still deal with Varroa as the predominant mite species in their colonies to deal with. So while Tropilaelaps is a slight problem in Korea, their primary concern is still Varroa mites. And so that is something that I would expect to see in areas of the US. Depending where you're located in the US, you might see that either Tropilaelaps, if you have brood year-round, is more of a problem, or Varroa mites remain consistently the problem that you have to deal with year-round if you're in the more temperate climates that experience a full brood break. So for the bottom line for our treatments, we know of effective treatments, but unfortunately, we do not have any treatments that exhibit that 100% efficacy, so we're still working on things and working with aphids to come up with an effective eradication plan in the event that we find Tropilaelaps somewhere. So with that, I'd really like to thank the Marl Honeybee Lab in Thailand and all of the research that I was able to do with them. I'd also like to ask you to please take advantage of all of our Tropilaelaps material that can be found on the Apiary Inspectors of America website. That's honeybeepest.org. On the top right, it has a Tropilaelaps tab. This has all of our most up-to-date Tropilaelaps information. And Ian, please feel free to share this out in any sort of newsletter. We try to keep this as up to date as possible. And so this is really kind of the cutting edge of where we're trying to update our distribution map. So as more things come out where this mite is spreading, we're trying to constantly update that to keep beekeepers as informed as possible. With that, I'd like to thank you. I'd like to thank all my funding. And then these are two of my publications. I believe the one on the left is the management study, and then the one on the middle, they're both published. That's my dispersal. They're both published open access. So if you want to read them, feel free, and the QR code should bring you to them. With that, I'd like to thank you so much for allowing me to talk to you and open it up for any questions. Thank you.
Thank you, Rogan. Yeah, we'll throw it out to questions, and if folks want to ask their questions, or if you want to put it in the chat, we'll make sure they get to Rogan.
Well, I liked it. Hi, Rogan. I'm Miguel. I'm with the Mount Diablo Bee Association, but I went to Man Lake on their sale, and there was a gentleman there that has a new mic control, and I was wondering if you heard about it. It's this one right here, Nora.
Okay, yes.
And I was kind of impressed by what I read, and I was just wondering your thoughts on it.
Yeah, so I think Noroa is one of the exciting things for Noroa. I just saw today. I'm pretty familiar and I'm friends with some of the people that kind of always are at the trade show for Noroa. It just got approved in the US, got EPA approval today. So that's really exciting for everybody at Greenlight Biosciences coming with Noroa. I think Noroa is extremely exciting. I think that it is a cutting edge, another tool in the beekeeper's toolkit. Um, I am interested to see if they're able to bring the cost down. I know that it is going to be rather expensive for varroa mites. For Tropilaelaps mites, it's going to be very interesting to see if it works for them. I know that they are interested and they're planning on doing hopefully some research to see if their product as it's currently constructed is effective against Tropilaelaps, because that would be awesome if you can do something that controls them both. But for varroa mites alone, I found that it should give you, a 3 to 4 month window of being an effective treatment and keeping those mites down. So as long as you have the mites at a low infestation at the beginning of when you put it on, then it should be hopefully effective long term in your colony.
Great, thank you.
I have a question. Is Tropy reducing the number of native colonies of the Asian honeybee and the giant honeybee, or do they coexist in equilibrium?
So that's a really good question. Thinking about, yeah, how they're coexisting. So they've kind of reached a form of equilibrium, the giant honeybee does. And so giant honeybee has a few different behaviors that it will do that assists in really preventing Tropilaelaps infestations from building up. One of the things that I didn't really have a whole lot of time to show you, though, is that Tropilaelaps mites for the Apis dorsata, they're better at pulling the mites off them. So if they do feel a mite, they have better grooming and hygienic behavior to remove that mite. The other important thing to think about is that Apis dorsata bees are open nesters. And so if they do remove a mite, that mite might be falling 6 or 7 stories. It's not crawling back up. Apis mellifera, it might fall down to the bottom board, and really no harm, no foul for that. The other interesting thing is that Apis dorsata is a much more migratory bee by nature. It will abscond. So if it reaches really high infestations of brood, it has no problem picking up and just moving to a new place and absconding and finding a new spot to live. This is another thing for Apis cerana, which is the Asian honeybee, they are notorious for. That's one of the reasons they're the original host believed to be for varroa mites. They have a few other mechanisms that help them control for varroa mites, but one of those things is they abscond very quickly. Of course, that's even more detrimental for Tropilaelaps mites because Tropilaelaps mites can't feed on those adults. So if they abscond, it takes a while for them to build up that brood nest. And so most of those mites are dying through starvation before they can get to larvae that they're able to feed on. And so they kind of have a little bit more of natural ways of preventing mite infestation compared to what we have. We don't want Apis mellifera. We don't want our bees to be absconding from our colonies and leaving. We want them to stay in there. We want them to produce a lot of brood, and we want them to produce a lot of honey. That's a little bit counterproductive then to Tropilaelaps, because that's what Tropilaelaps wants. They want a lot of brood in that colony so they can reproduce very easily.
Is there a lot of Western honeybees in that area? My experience up there, the hill tribe people seemed to all be honey hunters, and yeah, they catch an occasional swarm of the Serena and keep it for a little while if they could, but most of them were, like you said, taken off pretty quick.
So I did during my time meet one Apis. So there are no people that are actually, it's all honey hunters for Apis dorsata, the giant honeybee. Nobody can manage those. I did run into there's a couple people that do Apis cerana honeybee management, but they do have to deal with a lot more absconding from their bees comparatively to Apis mellifera. There are a decent amount of Western honeybee beekeepers there, but they don't have the number of colonies. So thinking about some of those, California commercial beekeepers, a lot like Sideliner beekeeper is kind of their commercial beekeeper level. So they're not going to be much over a couple hundred colonies maybe. And so that's another little bit different thing, is that they can do some more things and more intensive management with their colonies because they have less of them rather than trying to manage for some beekeepers upwards of 5, 10, even 20, 25,000 colonies.
Thank you.
Excellent. Well, thank you very much, Jamie. You stole my question. Uh, great talk, Rogan. Thank you very much. If there are no other questions, well, thank you. Thank you for joining us, even though there's the time difference.
Oh, yeah.
I really appreciate it, and we'll share out the information on the website that you mentioned, and pass it on to our members in our next newsletter as well.
Yeah, thank you so much for having me.